Thank you. I'm going to try to go quickly so as not to delay your lunch and have those vivid images fresh to our memory as we go to lunch. You heard about BioTime this morning from Leslie more generally. Today I'm going to talk to you about OrthoCyte, which is the orthopedic subsidiary of BioTime. What I'm going to try to do is give you a perspective on how we're positioned for making a great impact in regenerative medicine for orthopedics, especially because we have probably the broadest musculoskeletal progenitor stem cell portfolio in the industry, and we have also a highly adaptable biomaterials platform that enables our therapy development as well. So from a procedural perspective, we can enable our therapies. So OrthoCyte is focused initially on the aging spine for a very simple reason. The chronic back pain is a huge unmet need, and we think we're better positioned than most to address it in a unique way. And I'm going to tell you today about the challenge, what the opportunity is in chronic back pain, our solution with our osteochondral progenitors and our biomaterials delivery platform, and then how we're tailoring our approach. So chronic back pain is the largest health economic burden in modern society, and there's really two reasons for this. One is it affects everybody. So 85% lifetime prevalence, and it affects people in their productive lifetime. So as you can see on this slide, there's 3/4 of people with chronic back pain are between the ages of 18 and 64. So it's a huge unmet need. It affects everyone. Also, there's no treatment option. So for chronic back pain, typically it stems from the progressive degeneration of the intervertebral body and the intervertebral disc. Sorry, the disc is this small avascular cushion between the vertebral bodies, and allows your spine to bend and flex. And as the disc is progressively degenerating, there's really no therapeutic option, nothing that will slow down or reverse this degenerative process. So what happens is, as you go to see your doctor, if you have back pain, I'm not talking here about a twitch or a muscle spasm that will go away in a couple of days. I'm talking really here about chronic back pain that will sustain for a few months and longer. So as you see your doctor, the only thing that they can do really is manage the pain. And as the disc is progressively degenerating, ultimately you'll need most likely invasive surgery if you can't bear that pain. So really between surgery, invasive, risky surgery, and over-the-counter medicine, there's really not much that can be done. So at BioTime, we have developed a unique stem cell platform, and it's a little bit like this. It stems really from embryonic stem cell, but instead of using unpredictable embryonic stem cells, what we did is we nudged the cells through various differentiation protocols. We nudged the cell to differentiate just slightly, such that they acquire more tissue or more specialized characteristics and lose the unpredictability of the embryonic stem cells while preserving their highly proliferative potential. As you can imagine, we have to address large patient populations, so it's very important that our cells can be expanded to large commercial scale. So what we have specifically for the musculoskeletal branch of this tree, as you can see, we have now over 40 to 50 lines that are all different and can actually address all musculoskeletal indications. So some of these lines are good at forming tendon, bone, cartilage, and disc, etc., and you'll see why. So what we have is really enhanced attributes for all of these things. And I'm going to focus today on the potency and how we're utilizing this diversity and the ability to potentially select the cell line that matches best the indication that you're after. So this is what I'm talking about in terms of diversity here. I'm showing you 7 of our lines here. And like I said, some of these lines are really good at forming articular cartilage. Some are good at forming disc cartilage, tendon, bone, etc. And we're using these cells, and I'll show you how this translates into more real-life situation. What we did here is we isolated disc motion segments. So we can have intact disc that's bordered with a little piece of bone. We extract that from the spine of rabbits. And we induced a degenerative process in these discs, and then we inject our cells. We injected various cell lines that we have, and we compared this to a couple of cell line controls. And if we let this disc incubate in vitro for about 7 days, after that, what we can do is we can isolate this disc, and we can actually isolate separately the annulus fibrosus and the nucleus pulposus, which are the two main components of the disc, and look at molecularly what's happening, or how our cells are influencing this process. And if you see, if you inject MSCs in this disc, you can see that the cells will actually, you'll see the positive things of looking at, and you will see that you have induction of matrix component, that is, those proteins that are involved in repairing the disc. And at the same time, you're shutting down inflammatory modulators as well as matrix degrading enzymes. So this is what you want to see. And you can see with one of our lines, we have just about the same thing. Is one of our lines is really good at inducing cartilage, not cartilage, but matrix production and shutting down or repressing disc degeneration as well as inflammatory cytokines. But what's interesting is that some of our other lines can modulate the disc very differently. Like OTX5, for example, is really good at modulating very specifically just the nucleus, which is the gelatinous core of the nucleus. So it's really good at repairing very specifically the nucleus, whereas OTX3 is really good at reinforcing the fibrous structure of the intervertebral disc. And like I said, OTX1 actually can modulate both. So we don't really know exactly what this means when you translate this into the animal or into the clinic, but I'll tell you this, as soon as I can change the slide, that we're actually in a position to screen for this. So we can now look for, actually try these cell lines in vivo and see which one is going to be best suited for treating a degenerating disc in vivo. So this is now ongoing. The rabbit are ongoing right now, and we've injected these cells, again, various cell lines in the degenerating disc, and we can follow this by imaging with MRI and X-ray, and we can see the recovery of the repair process by histological and biochemical markers as well. So I'm going to present that probably later in the year. So the second element of our platform is this high-stem cell delivery system, and you've heard about this morning from Leslie. I'll just say this: it's derived from naturally occurring biomolecule, hyaluronic acid and collagen, and as you know, these components are ubiquitous. They're broadly used in medical applications. And for us, specifically in orthopedic, it really allows us to formulate various components in a liquid form, as Leslie was saying this morning, and then you can inject through a simple syringe or some simple delivery system and form a solid implant upon injection. So it allows the cells to stay in a confined environment and provide attachment factor for the cells to better engraft. So this is what we're doing. We're using this high-stem biomaterials platform in two ways. The first one, which is our near-term type of product development, we're combining this biomaterials with ceramic osteoconductive materials that allows us to form these bone grafting material that can be cleared through 510K. So this will allow us to launch product that are able to help bone heal, but obviously low potency type product. And then what we can do in the long run while we're doing this is continue the development of our cell-based products for induction of bone with osteogenic cells or osteoinductive cells, and disc repair with discogenic cells, if you will. So what we want to deliver in the next 2 to 3 years is this, using our biomaterials platform again, generate a series of product for bone grafting, different bone grafting solution in different geometries. These are not our products, by the way, these pictures, but just to illustrate the different types of geometries you can do for enabling orthopedic surgery and helping surgeon to heal bone. And then secondly, developing our cell-based product. We want to complete our proof of concept in animal these coming few months, demonstrating the utility of our cell-based approach in relevant animal model. Move the selected cell line, the one that we have screened that will allow us to pick the best suited cell line into a GMP compliant system and move into IND enabling work to hopefully get into first in human by 2016 for both early disc degeneration with our disc repair product and for late disc degeneration where the only thing you can do is really fuse the spine. So from a competitive perspective, if you look at cell therapies in general, I won't go through this table, but I'll tell you this. The difference between all of this and us at the bottom, and I'll grant you we're earlier than most, we're in preclinical stage. The difference is that we're not restricted with one cell type. We can actually, it's almost a pharma model where we can screen the cell that best address the indication that we're after, and in this case, we're interested in spine and disc degeneration. So now we have over 40 lines that we can select, and we can develop for various indications, including osteoarthritis and meniscus repair and all of these other orthopedic indications. So really in a position to screen and select. So from a partnering perspective, like I said, we're early, so we haven't partnered with anybody yet. But we're looking forward for various partnering solutions, and we're going to be looking for filling up this partnering matrix at some point. Then in conclusion, I'll tell you this. I think we're in a unique position to potentially address the entire care continuum of disc degeneration and chronic back pain. From very, very early on where the disc just starts to degenerate, you have the first event in back pain to potentially and hopefully slow down this disease process with lines that are able to specifically address this pathology, all the way down to when the disc is further degenerated and you need to actually regenerate or recover a new tissue with more potent lines, to ultimately actually inducing bone when the disc is so degenerated, it's actually rubbing bone against bone, and the only thing you can do is stabilize this segment with spinal fusion. So I think we're in a position to address this entire care continuum unlike anybody else. So I'll finish here, and I'll be happy to take questions on your way to lunch. Thank you.